Okay, on Monday, we left off, we were still talking about the cell, and we specifically started talking about energy creating organelles, right? So we'd sort of given an honorable mention to our peroxisome, right? Those little membranous sacs that use hydrogen peroxide to create an energy-causing reaction, an oxidation, to make ATP. And remember, the goal of all of these, I'm using yellow, okay, is ATP, which is the currency of the cell, right? So that's our big goal. So now we're going to take a closer look at the more effective, more efficient ways to make ATP in these cells, okay? The one we're probably most familiar with, and this is where we left off, was the mitochondria. Okay, and the reason the mitochondria, in part, is so much more effective, as we'll find too later in the semester, is because of these cristae. Okay, these many, many folds. Okay, so remember at the end of class, I akined all of these folds to like your kitchen having extra countertops. Okay, the more countertops you have, the more effective a cook you can be. Right, you have more surfaces to lay out your cooking. You can cool more cookies. Okay, so the more countertops you have, the more cooking you can do. It's the same thing here, right? The more folds that we have, the more surface area we have. Right, folds and countertops both mean surface area. It literally means space. You spit all over my mask. The more space we have to make that ATP. Okay, and so the making of ATP in this case is called cellular respiration, okay, which is the breaking down of that food source, usually a sugar, okay, all the way down into these usable currency bits. Okay, and we'll see all the details of that, I think, in like Unit 5 or 6. Okay, that feels good. That makes sense why the mitochondria is so good at what it does. Okay, so if we look down at this image, we can see all of these different folds. Okay, those folds, remember, are those cristae, all those countertops. Okay, we can also see why it's a membrane-bound organism, right? They're also membranes, making it harken to a eukaryote. I'm asking me a runny nose this morning. I'm sorry, guys. Particularly my online kids. You guys can really hear me doing that. Okay, so let's compare that to chloroplasts. So while mitochondria are found in both animal and plant cells, chloroplasts are, of course, found only in plant cells. Okay. Now, we are also extraordinarily effective at creating ATP. We just happen to use sunlight to do it, right? They cheat. So they don't need to get food. Okay. Now, instead of having cristae, many, many folds, okay, or chloroplasts do it with what are called thylakoids. Okay. Thylakoids are these little guys here. Okay, they're still a series of membranes, but instead of being folds, they're little stacks. I always think they look like little stacks of Girl Scout cookies, in particular Thin Mint. I'm a Thin Mint girl myself. Okay, but the idea is the same. Okay, by having these stacks of cookies, okay, that's lots and lots and lots of space. Okay, so we're still increasing the surface area by which we create that ATP. Still hitting myself in the face. I'm not getting any better at this every day. Okay, so just like those folds gave us countertop space to work on. Okay, these cookies, all right, these thylakoids are all surface area to make the ATP. Okay, the more surface area you have, right, the more countertops you have, the better you are at making ATP. Okay, so this is why chloroplasts and mitochondria are so good at their job, why peroxisomes really aren't as good at their job, right? So remember, peroxisomes was just a single membrane filled with fluid. So they can do it, but they just don't have the surface area that our chloroplasts and our mitochondria have. That feel good? Any questions about any of our energy-related organelles? Okay, but specifically for the most part, mitochondria and chloroplasts. Okay, so remember, we'll find mitochondria in all eukaryotic cells. Everybody needs a mitochondria. But it's the powerhouse of the cell. Okay, but chloroplasts are unique, right, in that they're a plant thing. It's a green organism thing. Okay, any questions so far? Okay, so the very last thing we need to talk about with cells, okay, is some structural components. Now, we're not going to get into a ton of detail here because we can get kind of lost in the woods. Okay, but we do want to mention that up to this point, we've sort of talked about how the cell has this cytoplasm in it, which is sort of akin to this vast ocean. And now we've stuck all of these organelles in it, and they just are what? Free floating? Which at its core is sort of problematic, right? Because in our journey, we said, well, we need to go from the nucleus to a ribosome to a Golgi body. And if everything's sort of free floating and loose in this ocean, how are you going to get from one place to another? And how in the daylight are you actually going to find everything if it's all moving around, right? So we have to have something that actually holds our cell together. We're going to anchor it and create these like roadways between one location to the next. So we call these components the cytoskeleton. Okay, so here again, we see that nice prefix, cyto, meaning cell. That's showing up right, okay. Right, and in this case, skeleton, right, just like we're thinking of, right, it's like a scaffolding. Okay, that underlying structure that's going to hold our whole cell together. Okay. So we've got a lot of things that need to be done in the cell, and we're just going to kind of lump these things together generally. Like I said, we really can get lost in the woods talking about the specific details of everything that gets done here. And these are things we will talk about in far more detail if you go on to take like a cell and molecular course. Okay, but generally, if we look at the functions, these are a lot of the things that we just mentioned. So if we think about anchoring organelles, okay, this is that idea that if we need to go to a ribosome, okay, that ribosome can't be free floating. Okay, it needs to be in one place so we can always find it. That's definitely the word one. Let's try writing that again. Okay, so literally like you think of boat anchor. Okay, it's going to kind of hold them down in one location. So as our transport vesicles are trucking around, they always literally know where they're going. Okay, because these things are set in place like little houses, more or less. Okay, if we consider these two, support in motility, okay, this harkens to the idea that we're going to build a roadway or a railway system. Okay, so we have a whole bunch of these guys that are going to cross-hatch across our cell. Okay, they're also going to create sort of like a suspension bridge structure, so it's going to help hold everything up, hold everything in place. Okay, going to help hold our plasma membrane out so it can't push in and collapse. Okay, and at the same time, these suspension bridges also create roadways that our transport vesicles can move across to get from the nucleus to the ribosome to the Golgi body. And just like our bridges are both structural and used to be traversed across. Or like a tunnel system, which is both structural and used for mobility. Okay, now ultimately, there are three major types of cytoskeleton features. I don't care that we can apply each, just know that there are three different types that are going to do these different jobs. Okay, so it's not just one type of molecule that is going to do all of these things, but just like with our biomolecules, you need a variety of molecules to do all of these jobs. My word keep kind of looks like it sucks, doesn't it? I think it's my E's. My E's are the problem. I can't write E's very well. Okay, this makes sense, sort of what we're doing here. Okay, so if we take a look at this, okay, at the bottom we have the three different types. And again, I don't care that you memorize which type does what, but in our image, and this is sort of what we want to pay attention to. For example, this is a mitochondria, and we can recognize that. We see that folded cristae inside. Okay, we can see that that's being anchored in place. Okay, so mitochondria is not just free-floating in the ocean, which we can't see, right? We've taken the cytoplasm out. So everything is empty space. We can actually see what's going on. So it's being held in place, so it's not going to free float around. Mercy, that's hard to say. Okay? We can also see our roadways. Okay, things on which we can move along to get from one place to another. Okay, so we can start to see how these things are working together to create both structure and roads to get to, say, our mitochondria, or what I suspect is the rough ER, right, because it's got all those dots on them, which is probably ribosomes. None of which matters, right? The idea is that we're just seeing how our organelles are being held in place and how you can now feasibly find them in what would be a large cell if you were a tiny transport vesicle. Right, any questions about what's going on with the cytoskeleton? Okay, so broad strokes with this, we just kind of want to understand what the purpose of the cytoskeleton is. Shiny? Silence? Too early? Okay, I'm actually going to skip this and we're just going to ignore it for now. Alright, so let's do our in-class activity. Okay, so just like last time, do what you do. Okay, we're going to draw this time, so depending on your computer, that may affect what you do. Okay, if you are able to draw on your computers, you can use PowerPoint or Paint. I don't care. Right? These don't have to be beautiful. If you have a touch screen, you can also draw on there too. Okay, so that's fine. You can also use a piece of paper though, I don't care. Just make sure that you're taking a picture and it's very clear. Okay, so here's the key. Okay, without using your notes to the best of your ability. Okay, you're going to draw me a protein being made, following all of the things that we've done in class. So where do we start? What do we need? Okay, what are all of the organelles that we need to do this? Okay, from start to make to leaving the cell, which is something you wouldn't normally do, but that's what we're going to do here. Okay, so we can use as many different organelles as possible. Okay. Also note that our cell needs to be not dead. So we need at least one energy organelle and one structural component. Okay, please do not make these the world's tiniest drawings. We have to be able to see them. And label your organelles. Okay, don't use your notes for this. Okay, try it first without. Okay, remember we're using these as study tools, so now you're practicing your own memory recall. Okay, so let's take 5 or so minutes to give this a shot. In the meantime, I can open the Dropbox. Nope, that's not what I want. Okay, any major questions about this? Taking notes on my. Okay, so some quick reminders before we switch gears then. One, you can quit recording this session.